Hello and welcome to Lecture 2. We're going to be talking about the resting membrane potential here and the movement of ions a little bit. We'll also touch on the action potential at the end, but we'll cover that in greater detail in Lecture 3. I want to set up what membrane potential is and the source of membrane potential. Then I want to talk about equilibrium potentials or reversal potentials. They're the same thing and they're absolutely fundamental for understanding the resting membrane potential, but then also how any ion channel will affect the membrane potential. If you understand equilibrium potential, you'll know a lot about the electrical activity that goes on in nerve cells. Okay, so first let's talk about membrane potential. It's exactly what it sounds like. We've all learned about potentials in our basic physics classes, and a potential is just a separation of charge. And so a membrane potential then is a separation of charge across the cell membrane. Every cell has a membrane potential. And the reason for this is because we have a different amount of ions outside and inside the cell. At least specific ions are going to have different concentrations inside and out. The membrane potential of your textbook resting neuron is negative 70 millivolts. This is largely what you'll see in your neuroscience classes. You won't find exactly negative 70 millivolts in really any of the neurons that you go out and measure in the real world, but they're pretty close. They're in the ballpark. Some of them are a little less, some of them are a little more than negative 70 millivolts. And this difference in charge has everything to do with the separation of ions across the membrane. Certain ions are going to be at higher concentrations inside the cell. In this little table here on the left, you can see that potassium, for example, has a greater concentration inside than out. On the other hand, sodium and chloride are found in greater concentrations outside the cell than inside. And this has to do with our evolutionary origins, where we came about, by we, here I'm talking about the very first cells that developed, certainly not humans. The very first cells that developed came about in a saltwater environment. And so the extracellular fluid would essentially just be salt water. The inside of the cell then distinguishes itself by creating a different ionic balance. The concentration of calcium is very low on the inside. That has everything to do with the high concentration of phosphates. They're impermeable anions, and we'll touch on those in a second. I don't want to get ahead of myself. But you can see these ionic balances shown on the illustration on the right there. But essentially, those separations of charge are going to help establish the negative membrane potential, and they're also going to be used when neurons fluctuate their membrane potential. Now, even though we have this difference in specific ions where there's more potassium inside, but there's more sodium and chloride outside, there's still equivalent osmolarities inside and outside the cell. In other words, there's just as much stuff dissolved inside and out. Because if there were more ions inside the cell, then water would come in, the cell would swell up and burst. And if there were more ions outside the cell, then water would leave and the cells would shrivel. So we have to maintain that equal osmolarity, even though we've created these differences in individual ions. Okay, just keep in mind, we don't have differences in the total amount of dissolved substances. All right, we just create these differences in specific ions, and that's what creates our membrane potential, which we can measure using a technique called patch clamp recording. And in patch clamp recording, you'll take a little piece of glass. That's the recording pipette shown here. There's our little cartoon neuron in the upper illustration. If you zoom in, what they've done here is make contact with the membrane, and then they apply a very mild suction. So just a little bit of negative pressure to cause the membrane to kind of hug up to the glass there. Now this is just a flame-polished, very small pipette, so it has a nice smooth surface and it won't poke through the cell membrane there. Now you can remove this little portion of the membrane by simply pulling the pipette back, and now you've isolated, in an ideal world, one single channel to study, but really you're going to get a few. Or you can apply a little stronger negative pressure in your pipette, you can create a little harder suction that's going to pull open a portion of the membrane, and now you can listen to the activity of all the ion channels in the cell. So whole cell recording is what we call that. This is very useful whenever you want to have clean recordings of ionic currents or the membrane potential. If you just simply want to listen to gross changes without breaking open a hole in your cell, you can remain cell attached. That's what they're showing on the left there. Once you've pulled open a hole and gone whole cell, you can still pull back your pipette, and if the stars align, you'll pull off a little bit of membrane and, in an ideal world, a single ion channel here. But what you'll notice is that it flips its orientation. Because the two flaps of membrane there don't like to stay free floating because of that hydrophobic core. They quickly seal back up on one another, and now you have an ion channel with the extracellular face exposed. This makes it much easier to apply ligands like neurotransmitter because you can just simply put your pipette into a bath that contains your neurotransmitter or an inhibitor, and you can see how the ion currents change. So the inside-out recordings are useful if you want to study molecules that bind to the intracellular face of your ion channel. Outside-out recordings are really much more useful because they expose the extracellular face of your ion channel, which is typically where your ligands will be binding. Now, if we were to measure the membrane potential of a neuron, we'd find that it's negative, as is really every cell in our body. The textbook value would be -70. Just keep in mind that it fluctuates a bit. The reason that we have this negative potential is in part because of impermeable anions. Those would be phosphates. That's that PO4. Phosphate groups have a negative charge, and you find phosphate groups in all of our nucleic acids, so all the DNA and RNA in our cells. Also ATP, which is kind of floating all throughout our cells. That's our source of energy. It's also a part of RNA there. That also contains phosphates. You'll also find phosphate groups floating around freely by themselves and attached to proteins as well. And we call them impermeable anions because they don't permeate the membrane. They never leave the cell. And they're anions because they have a negative charge. So because the cell is filled up with these phosphates, that biases it toward a negative membrane potential. It also requires that calcium levels remain low because calcium phosphate comes out of solution very easily. In addition to those impermeable anions, at rest, the cell is largely going to be permeable to potassium, so it's always leaking potassium. Now, if we were to recall our relative concentrations of potassium, we'd remember that there's a lot more of it inside than out. And so the diffusive force is going to push potassium out. Potassium has a positive charge. Because positive charge is leaving, that's generating a negative current and thus creating that negative membrane potential. Now, of course, because we're always leaking ions, this must be actively maintained, and that's going to be the job of the sodium-potassium ATPase, which we will focus on more in Lecture 3. Now we're going to give you -70 as the textbook value for neurons, but other cells are going to have a resting membrane potential as well, and different cell types can have different resting membrane potentials. Those astrocytes, those glial cells that we discussed in Lecture 1, those tend to rest more hyperpolarized than neurons, so they're usually around -80, but it totally depends on the astrocyte. Some actually rest around -60. And the photoreceptors in our eyes that pick up light, those rest around -40. So different cells will have different resting membrane potentials, and as we are about to find out, that's because of differences in their membrane permeabilities. Now, before we can talk more about that, we need to understand how ions actually move through the membrane. And they're going to do that through proteins. For the most part, it's going to be through channels, but there are also some transporter proteins that are very important. When ions are flowing through those channels, there's going to be two forces that act on them. There's the electrical force and there's the diffusive force. They're going to flow until they reach their reversal potential. That might not make a whole lot of sense now, but it will as soon as we step through this. Now ions, because they are charged, cannot freely move through the membrane. There's that hydrophobic core that prevents hydrophilic molecules like ions from moving through it. So we have to have some sort of an aqueous pore that the ions can move through. Now ion channels are going to have an open pore, so this is just free movement. It's going to be diffusive and electrical forces pushing ions through the membrane. It's totally passive. In passive transport, the ions are going to move according to their electrochemical gradients. We don't need any energy because we're essentially using the potential energy that exists from those concentration gradients. For example, potassium leaks out of the neuron. This doesn't cost anything. This is free. Leak channels are open and potassium leaves because there's a whole lot more of it inside than outside. Active transport, on the other hand, uses cellular energy, which is ATP, to create the ionic imbalance. So active transport uses transporter proteins, also called pumps, to move ions against their concentration gradient. We're not going to touch on these in great detail here, but we will bring up the sodium-potassium ATPase. And its name tells you what it is. It moves sodium and potassium using ATP. So if ions are moving with their concentration gradient, it's passive transport. It's totally free. If they're moving against their concentration gradient, this is active transport because it requires cellular energy. Both of these are going to be mediated by proteins. Again, ions can't pass freely through the membrane. They have to move through a protein, either a channel or a transporter of some sort. Now, whenever we open up ion channels and ions start moving across the membrane, that's going to change our membrane potential. Some ion channels are going to just leak, like those potassium leak channels, and others require some sort of a stimulus. It might be an environmental stimulus like pressure or stretch. So for example, if you were to press on your fingertip, as is going on in this cartoon, there are special receptors there to sense the pressure, and then they pull open ion channels with the mechanical force of the pressure. So by pushing down, that stretches cell membranes, and it actually physically pulls open those aqueous pores, and ions move through them. And what you get is a change in the membrane potential. So when you touch the skin, those neurons that are receptive to touch are going to depolarize a bit. Other ion channels are going to be sensitive to neurotransmitters, so they'll need a chemical stimulus rather than a physical stimulus. In this cartoon, what they're doing is stimulating that blue axon, which is going to release neurotransmitter, and that's going to bind to neurotransmitter receptors on the dendrite of that cell that they're recording from, and you can see that this must be an excitatory synapse because they get that depolarization. So they're probably releasing glutamate from that blue axon. So it's not physical stretch. We're not physically pulling open the ion channel. In this case, the neurotransmitters are a chemical signal that biases the ion channel toward opening. And then there are some ion channels that open in response to changes in membrane potential. These are going to be the ion channels that are responsible for action potentials and active signals like that. So suppose that we were to dump even more glutamate onto our neuron. Rather than depolarizing a little bit, what you'll find is that that little bit of depolarization activates voltage-dependent ion channels, and then they open up to create this very robust depolarization where we go from a negative membrane potential to an actual positive and then back, all within the span of about 3 to 4 milliseconds. So those action potentials, that's what we're looking at here, that's an action potential. Those are the active signals that neurons are going to generate. They're absolutely voltage-dependent signals. You have to cross that threshold, okay? So what we're looking at here is the membrane potential of a neuron as we inject different amounts of current. Okay, so if you inject an electrical current, you will very rapidly change the membrane potential of a neuron. So they have their stimulating electrode and the recording electrode on the same neuron, and the upper portion there in part B, that's showing you the injected current. 0 means they're not injecting any current. When it goes down, that means they're injecting a negative current, which, as you can see below, translates to a negative change in the membrane potential, thus a hyperpolarization. Whenever the current is positive, as we can see in the third step up, then the membrane potential becomes depolarized. With more positive current injections, you can see that you start to generate those action potentials. But as you increase the strength of your current, you don't get any more positive potential out of it. Instead, what you get is a greater number of action potentials, and thus we say that action potentials are all or none. We'll see this again at the end of this lecture. No matter what you do to open up your ion channel, once it's open, ions are going to move in accord with two forces that act on them. The first is the diffusive force. Okay, this is based on the concentration gradients that we discussed earlier. So potassium will want to flow out of the cell based on its concentration gradient. Sodium, on the other hand, will want to flow into the cell. That's because there's a lot more sodium outside than inside, and so as it moves randomly and spreads out, just by chance it's going to flow into the cell. But there's another force that acts on these ions. Because they have a charge, the electromotive force is also going to determine how they move. Sodium is a very simple ion. There's a lot more of it outside, so it wants to go in. The inside of the cell also has a negative charge, at least at rest. So that negative charge further attracts sodium to come into the cell. Potassium doesn't all leak out of the cell because the cell has a negative potential. And in fact, if the cell becomes negative enough, we can actually have potassium enter the cell. So potassium is kind of torn. In one respect, it wants to leave because there's a lot more of it inside. But on the other hand, it's a negative environment, and so the positive charge of potassium is attracted to the inside of the cell. You can very easily determine whether an ion is going to move in or out with a very simple equation, which we will get to later on, called the Nernst equation. But these ions are going to flow until these two forces even out. Okay, so we have to have the diffusive force and the electromotive force being equal, but in opposite directions. Now, as ions are moving out or into the cell, you might expect that their concentration is going to change, but they really don't. Okay, let's have a quick reality check here. So you don't really get appreciable movement of ions anything beyond about 4 nanometers from the membrane on either side. And the typical cells that we're dealing with are going to have at least a 15 micron diameter, if not larger. It's not uncommon for neurons to have 20 micron diameters. And this is just neglecting the dendrites and the axons. In the cell body, if you look at what 4 nanometers equates to, let's look at the illustration on the left there in part A. That box is a zoom in of the lipid bilayer there, showing you in red where we should have appreciable movements of ion. And you can see that this decays very quickly as a function of distance. That's shown in part B. Okay. While that's illustrative, we can get a lot out of part A too. Now, if you look at the bottom, this illustration still has that little red part, but what you'll notice is that you can't really see it, and that's because the actual volume affected by ion flux is negligible. We're dealing with fractions of a percent. So we should assume then that as ions are moving across the membrane, that the diffusive force is constant. This is essentially going to be constant because we're dealing with very small fractions of the volume of the cell, and also we have that active transport. Okay, as potassium leaks out, or if sodium flows in, this gets offset by those ion pumps. In this case, the sodium-potassium ATPase. We spend a molecule of ATP to remove 3 sodiums and bring in 2 potassium ions. So as potassium leaks out, we're spending ATP to bring it right back in. Okay, so because of that, because the ions are really only moving within 4 nanometers of the membrane, and we have active transport, what we should assume is that our diffusive force never changes. The only thing that's going to affect us then is that electromotive force. So it's only changes in the membrane potential that affect the movement of ions. So whenever we're talking about our electrochemical forces canceling each other out, what we're saying is that ions are going to flow until they reach the membrane potential that then offsets their diffusive force, because diffusive force is always constant. And that potential is what we call the equilibrium or the reversal potential. And both of those names make sense. It's called the equilibrium potential because your ions are essentially at equilibrium. There's no net movement. They're not at equal concentrations. They're at equilibrium. There's still, in this cartoon here, way more sodium outside the cell than inside, but because the cell is so positive, that repels the sodium as strongly as the diffusive force is pushing it in. The reversal potential also makes sense because now, instead of getting an inward flow of sodium at the reversal potential, it actually starts to reverse. So there's sodium flowing in, there's some sodium flowing out, and it's moving at equal rates. At the equilibrium potential, if the ion channel is open, ions are still flowing. There's just no net flow. There's just as much coming in as there is out. So let's walk through this illustration on the right there and see if this makes sense. So in part A, we're at rest. All right, our membrane potential there is negative 70 millivolts. We're only going to look at the movement of sodium here, right? Because it has very clear direction. At rest, the diffusive force is pushing us inward. That's what that blue arrow there on the right is showing. Diffusion is saying go in. That's because there's way more sodium outside than inside. The electrical force is also saying go in because it's negative. At negative membrane potentials, positive ions want to come inside. So sodium flows in. Now when that positive charge is entering the cell, right at the vicinity of the membrane, that's going to change the membrane potential. That changes the charge across the membrane. So after a little bit of time, let's say it reaches 0 millivolts. It's going to go beyond that. But at 0 millivolts, now let's have a look at our two forces. Remember, diffusion's constant. We have those pumps moving, and we're not really moving an appreciable amount of ions. Okay, remember, just a small fraction. But the electrical force has certainly changed. If we have a membrane potential of 0 millivolts, that means that the sodium is neither attracted nor repelled by the membrane potential. So there's really no electrical force at this point. It's all diffusive, but it's still being pushed inside. So sodium will continue to flow in until it reaches the reversal potential. And that would be positive 56 millivolts, somewhere in that ballpark. At that point, we have a positive membrane potential. It's going to repel sodium. And at positive 56, it repels sodium just as much as the diffusive force pushes it in. So when we're a little less than 56, like if we're at 50 millivolts, well, the diffusive force is still stronger and sodium still flows in, just weakly. If we're above that, if we happen to go to, let's say, positive 70 millivolts, now the electrical force will be stronger than the diffusive force and sodium will flow out of the cell. So once you open a channel, these two forces are going to move the ion until the diffusive and electrical forces cancel out, and that's our equilibrium or reversal potential. And we can calculate that by using the Nernst equation. This is a very simple equation as long as you make the simple assumption that we're at body temperature. And if you're alive, you're functioning at body temperature. So the reversal potential is really just based on the ion concentrations, which are essentially constant. So the reversal potential is essentially constant. So the entire equation is shown at top. If we go ahead and substitute the constants and then assume body temperature, we can simplify this equation to be 61.5 divided by the charge of the ion. So if it's sodium, we divide by 1. Very simple math. If it's chloride, we divide by -1. And then we multiply that by the log base 10 of the fraction of the ion outside over the concentration of the ion inside. So if you were to plug and chug those values from early on, you'd find the reversal potential for potassium is -102, The reversal potential for sodium is positive 56, chloride is negative 76, and calcium is positive 125. And they're plotted there on the bottom, just to give you an idea of where they stand relative to the resting potential and 0 millivolts. So you can see the resting potential is very, very close to the reversal potential for chloride. It's more positive than the reversal potential for potassium. So if we were to open a potassium channel up at V rest, then we would move toward the reverse potential for potassium, so potassium would flow out. If we open up its sodium channel, sodium flows in. Right? We can tell all this because we know the reversal potential. If you know the reversal potential of an ion, you can predict its behavior. It's very simple. So let's say we'll look at chloride. That's our anion, shown in kind of an orange sherbet color there. You can tell which direction that ions are going to flow. So if the membrane potential is more negative than the reversal potential. So the membrane potential is shown on the x-axis, and the movement of ions is on the y-axis there. If we're more negative than the reversal potential, then cations are going to enter the cell, right? Because it's a negative charge. Cations have a positive charge, and they're attracted to that. Anions will leave the cell if we're more negative than their reversal potential, because the negative charge will repel the anions. Conversely, if the membrane potential is more positive than the reversal potential, then cations will leave and anions will enter. For a real-life example, if we're dealing with potassium, resting membrane potential is negative 70, but the reversal is negative 102. Therefore, the reversal potential is going to be more negative than the membrane potential. And so since our membrane potential is more positive, that means that potassium is going to leave the cell. On the other hand, if we're dealing with sodium, the reversal potential is about positive 56. At rest, we're at negative 70. Because the membrane potential is more negative than the reversal potential, then that means that sodium will want to enter the cell. We can also tell from the reversal potential the net flow of an ion. We can calculate what we call the driving force, and that's really just the difference between the membrane potential and the reversal potential. And the larger these are, the greater the movement of ions. At reversal potential, the net flow is 0. There's no driving force. So if the membrane potential is positive 56 millivolts and we open a sodium channel, we'll have no net flux of sodium. We're already at the reversal potential. On the other hand, at resting membrane potential, at that negative membrane potential, there is absolutely a driving force, because negative 70 minus positive 56 is going to give us negative 126. Okay, so we have a driving force here to cause sodium to move into the cell. You can tell this by looking at this little XY plot. So let's look at our purple line there, the cation. That's sodium. It reverses at positive 56. As you move more negative on the x-axis there, so as you move to the left, you'll notice that we're moving further away from our x-axis. In other words, the y value is getting much greater in the negative direction, and if we were to move more positive than our reversal potential, then we're moving upward along the y-axis as well. So we're creating a greater movement of ions. And you really do see this linear relationship in the real world. So if you understand the reversal potential, and you can calculate it using the Nernst equation there, or if you just simply remember the values that I've given you here, you can tell how ions move. We should know that if we're more positive than the reversal potential for a cation, well, then that cation is going to leave. So at rest, potassium leaves. At rest, sodium enters, and that's because of differences in their reversal potential. Okay, so if we understand reversal potential, now we're ready to talk some more specifics. So the membrane potential is going to be determined by multiple ions, okay, not just one. So you can use the Nernst equation to figure out the reversal potential for individual ions, but in the real world, Neurons are permeable to more than just one ion at a time. So let's start off with the resting membrane potential. VREST. This is going to be established by leak channels. By definition, a resting neuron isn't doing anything, so the ion channels that are open here are always open. They don't need any stimulus to open, so they're what we call leak channels because they're leaky. There's always this leak of ions through them. Thus, the resting membrane potential has to be determined by leak channels. We can't be dependent on a signal or we're not at rest. But what we should know is that we must be selective for certain ions, otherwise we'd expect our resting membrane potential to be somewhere around 0 millivolts. If any old ion can move through a leak channel, we shouldn't really generate any net current. So this clearly can't be the case. We must have some selectivity for specific ions. Here's another equation. This is essentially a weighted Nernst equation, where we just put together multiple ions based on their permeabilities. That's all this is. This is called the Goldman-Hodgkin-Katz equation, and this can be used to determine the resting membrane potential or the membrane potential based on any permeability for these different ions. So if you were to plug and chug the concentrations of sodium, potassium, and chloride inside and outside the cell, as well as their relative permeabilities, what you should find is that the resting membrane potential should be somewhere around negative 73 millivolts with textbook values. Now the relative permeabilities we of course determined empirically. We call potassium 1, okay, that's the one that we're the most permeable to. So relatively speaking, the permeability to sodium is only 4% that of potassium. So we're far more permeable to potassium at rest than we are sodium. This is good. This allows the cell to have that negative membrane potential. If we're more permeable to sodium, well, then we wouldn't be as negative. We'd move toward the reversal potential of sodium. Chloride, you might expect to actually be the most permeable at rest because the reversal potential of chloride is so close to the resting membrane potential, but it's not. We're about half permeable, or 45% permeable to chloride compared to potassium. So we're most permeable to potassium. There's a fair amount of chloride permeability and very, very low sodium permeability. You should be able to use the Goldman-Hodgkin-Katz equation to calculate membrane potentials as permeabilities change, or as ion concentrations change. Okay, it's a very simple equation, and you can even set up a spreadsheet to do the calculations for you. But this is what we use to calculate expected membrane potentials. And this gives us our resting potential, and it tells us that we're most permeable to potassium, so that leak is largely a potassium leak. There's also a bit of a chloride leak, and there's very little sodium leak. There is some, but it's negligible for the most part. Because we have this permeability to all these ions at rest, none of them are zero. And because of that, the cell is going to be sensitive to both increases and decreases in the permeability of each ion. Okay, this allows the neuron to carry out more complicated signaling because now it can receive an increase or a decrease. Right? There's bidirectional signaling possible. But for the most part, the signals that it gets are going to be very simple. There's going to be depolarization that leads to the action potential, or there's not going to be sufficient depolarization and the neuron stays at rest. Okay, so neurons to some degree are binary. That action potential is nothing more than very rapid depolarization followed by repolarization, a very brief hyperpolarization, and then we're back to resting membrane potential. And that's going to move on down the axon. We'll talk a whole lot more about the action potential in Lecture 3, but I want to touch on it quickly here since it was covered in the chapter. Now, knowing the reversal potential is very powerful because anytime you increase the permeability of the neuron for that ion, membrane potential always moves toward that ion's reversal potential. Okay, so if you go and plug in different permeabilities in your Goldman-Hodgkin-Katz equation, if you were to increase the permeability for sodium, you'd find that the membrane potential is going to change. It's going to move toward the reversal potential of sodium. So if we're resting at negative 70, let's say that we increase our potassium permeability. So if we open up more potassium channels, what's that going to do to the membrane potential? We should know this, because we know we know that the reversal potential for potassium is somewhere around minus 102 millivolts, and it's plotted down there on the bottom. So if we're resting at minus 70 and we open potassium channels, we're going to move the membrane potential toward the reversal potential of potassium. So if we were to reach maximal potassium permeability, we would actually bring the membrane potential to negative 102 millivolts. We wouldn't go any more negative than that, it's impossible, but we would bring it to the reversal potential. On the other hand, let's say that we increase our sodium permeability instead of increasing potassium. Well, if you have a look, you'll see that the reversal potential for sodium is far more positive than our resting value of minus 70. As we increase permeability for an ion, the membrane potential moves toward its reversal potential. It never goes above it or below it, depending on the ion. So what we're going to do, if we have maximal sodium permeability, we're going to actually bring our membrane potential to positive 56 millivolts. We can never go above that. Sodium can't bring us to positive 57 or 58 millivolts, because once it hits the reversal potential, those two forces, the diffusive and the electrical, cancel each other out. This is a very important concept. You must understand that when you increase the permeability for a specific ion, the membrane potential always moves toward that ion's reversal potential. For example, if we increase our calcium permeability, that could bring us up above positive 56 millivolts and then bring us toward the reversal of calcium. When we increase our chloride permeability, that will give us a very slight hyperpolarization. It'll make us a little more negative. This is going to be very important as we work through the action potential, because active neurons are going to be moving a lot of ions around, and we want to understand how the movement of ions works. And if you understand reversal potentials, you got it. So let's set the stage here. The action potential, for the most part, is going to be caused by that synaptic input, and we're going to think of our synapses as being between the axon and the dendrites. Okay, that's what we're seeing here. We're seeing little ion currents being generated in the dendrites. All right, those are going to funnel into the cell body. Okay, so we can see here that those dendritic ion currents, they're going to cause depolarization there. And in the cell body, if we depolarize sufficiently, remember, we're going to generate action potentials. Okay, so the cell's going to fire one or several action potentials. All right, now how does this work? Let's go through the action potential. All right, on the top cartoon there, this is just setting the stage. We're at rest, and at rest, it can only be leak channels. So only the leak channels, and for the most part, those are potassium channels. There's some chloride leak channels, and there's very few sodium leak channels. So we'll just think of it as potassium for simplicity's sake. If the neuron is sufficiently depolarized by that synaptic input, it's going to reach what we call threshold potential. This is the potential, so it's the charge across the membrane. That's the threshold for firing an action potential. If we were to reach threshold potential, then the neuron is going to depolarize substantially, and that's because of an increase in sodium permeability. We have what we call voltage-gated sodium channels, and they're exactly what they sound like. We should think of the membrane as a fence, and these ion channels are kind of like gates. Now, what causes the gate to be open? Well, a leak channel is just a busted gate. There really is no gate. It's always open. It's an opening in the fence. Voltage-gated sodium channels, on the other hand, have a gate, and that gate gets open depending on the voltage, depending on the membrane potential. Now they're sodium channels. That means that they allow sodium to move through them, not potassium. Now remember, at rest or even at threshold potential, we have a negative membrane potential. There's a lot more sodium outside the cell than in, so the diffusive and the electrical forces are going to push sodium in until it reaches its reversal potential. Or in this case, until the voltage-gated sodium channels inactivate. We can see that these channels actually turn themselves off. Okay, so after about a millisecond or so, the neuron is going to quickly repolarize. All right, rather than depolarize and get that positive membrane potential, the voltage-gated sodium channels are going to inactivate themselves. So they're going to turn themselves off. Now our sodium permeability is back down to that 0.04, the very, very low value. But we've opened up another set of potassium channels, the voltage-gated potassium channels. Notice that they're not leaks. They are gated by membrane potential. So they're activated by the same threshold value as voltage-gated sodium channels. They're just much slower. So they're 1 millisecond slower, which is much slower in the case of ion channels. So they open a little bit after voltage-gated sodium channels. And when they do, we're at a positive membrane potential, so we're very far from potassium's reversal. Potassium really, really wants to leave the cell. It has a positive charge and there's a bunch of potassium inside. Now what you'll notice is that we're going to actually bring the membrane potential lower than that resting value, and this is what we call the after hyperpolarization. And that's because our potassium permeability is higher than at rest. We have not only our leak channels open, but we also have those voltage-gated potassium channels open. So the relative permeability to sodium is much higher, and as you increase the permeability for an ion, you move the membrane potential closer to its reversal. So we don't just stop at rest, we don't stop at -70. Because we have that increase in potassium permeability, the membrane potential should be more negative. It should be closer to potassium's reversal potential. And if you were to increase that permeability value in the Goldman-Hodgkin-Katz equation, you'll find that the membrane potential does indeed begin to approach potassium's reversal. That's what gives us that after hyperpolarization. Now, because we're back at a negative value, that voltage gate closes. So we go back to our resting permeability, so it's only the leak channels, and because of that, we return then to our resting membrane potential. So we've reduced our potassium conductance back to its normal resting value. And so that after hyperpolarization is very brief. All right, it's only until our voltage-gated potassium channels all close and we return back to resting conductances. And that's it. That's the action potential. It's very simple. It's about 3 to 4 milliseconds long, and it's actually going to keep itself going because it's activated by depolarization, and then it causes substantial depolarization. We're going to have a better look at this in Lecture 3. But this is just the most exciting 3 to 4 milliseconds in a neuron's life. So you can remember what goes on in an action potential if you break it down into just about 1 millisecond at a time. In the first millisecond or so, that's where we have our strong depolarization. So there's that increase in sodium conductance. In the next millisecond, we have our repolarization. That's where we inactivate our sodium channels, and we have the opening of our voltage-gated potassium channels. That increase in potassium conductance leads to that final millisecond, the after hyperpolarization. Once the voltage-gated potassium channels close because they're below threshold, we're going to return back to rest. Now you're going to hear me say depolarize, you're going to hear me say hyperpolarize, and you'll hear repolarize in the action potential. What those have to do with is removing that membrane potential, really. So depolarization is really just going to refer to a positive change in membrane potential. That's it. If we're moving positively away from resting membrane potential, it's depolarization. That's always the case. So if you're going from negative 70 to negative 70 to negative 65 millivolts, depolarization. Negative 70 to negative 60, depolarization. If you're going from 0 millivolts to 10 millivolts, it's still depolarization. Repolarization is going to be a negative change toward Vrest. So if you're going from 0 millivolts to negative 10, that's repolarization. But a negative movement isn't always repolarization. If you're going from negative 70 to negative 80, that's hyperpolarization because you're even more negative than you were at rest. Okay, just a couple terms to keep in mind. Really, the ones you need to know would be depolarization and hyperpolarization. Depolarization is positive relative to rest, and hyperpolarization is negative relative to rest. Now, just as a quick note before we get an in-depth discussion of action potentials, because of how they're generated, they are essentially all or none. This isn't always the case in real life. There are action potentials that actually diminish in their amplitude, but as a first pass, we should think of action potentials as all or none, because that increase in sodium conductance should bring us towards sodium's reversal every single time. And it should be consistent, and usually it is. Okay, so what we're looking at here are recordings from a neuron that are given different amounts of current injection. Okay, so there's three things going on here. In the top one, they've injected 160 picoamps, so a very small amount of current, just below threshold. They add a mere 2 more picoamps, so 162 picoamps. And you can see that it's going to fire several action potentials at a fairly constant rate. It starts to attenuate a bit, but it's fairly constant. When they add even more current, you don't get an increase in the height of the action potential. It's not depolarizing anymore. Instead, what it's doing is generating a greater frequency of action potentials. So injecting more current is analogous to a more intense stimulus. So brighter light or more intense pressure on your fingertip is going to generate a greater frequency of action potentials. You won't get larger action potentials, you'll just get a lot more of them. And this is what we call frequency encoding. And as we'll find out, the whole purpose of generating an action potential is to release neurotransmitter, to release those chemical signals that get passed between neurons. So a more intense stimulus leads to more action potentials, which leads to more neurotransmitter release. Greater depolarization has nothing to do with it. All right, it's all about the frequency of action potentials. We'll talk a whole lot more about action potentials and the voltage-gated ion channels that create them. The purpose of this lecture is to introduce you to ion channels, membrane potential, and importantly, reversal potentials. Make sure you're comfortable working with the Nernst equation and the Goldman-Hodgkin-Katz equation. Send me any questions you have. I look forward to seeing you for Lecture 3.